Hey Mustangs, in this video we're going to take a quick look at exocytosis. So, in order to do this, you're going to need your handy dandy exocytosis handout that you received in class, so that way you can write all the information down in one place. Alright, let's go ahead and get started. So, exocytosis is going to be the opposite of endocytosis. So in endocytosis, we saw that if you wanted to get large quantities of small molecules in or really big molecules in that didn't fit through the lipids of the plasma membrane or couldn't go through the transport proteins of the plasma membrane, then endocytosis was the way to get them in. Here in exocytosis, the key is exo for out. So endo brought molecules in, exo is going to export them out of the cell. So think exo for exit. So exocytosis is a type of active transport that uses ATP energy to transfer large molecules or many small molecules out of the cell using vesicles. So the key here is the fact that it is a type of active transport. So we know that energy is going to be involved. So let's go ahead, go down here to our illustration and take a look at what's going on with exocytosis. So just like with endocytosis, we're going to show it as a series of 3 steps, but now this time exiting the cell. So we're going to start here with step 1. So this is the start of the image. Now remember, this is time lapse. This isn't all happening all at the same time. Okay? So we're saying this happens first, and if you look at it, let's say a few minutes later, just to wrap our minds around the timing here, a few minutes later you would see this, and then a few minutes later you would see this. Okay, so a series of steps here, not all at once, but first, second, third. Okay? So in our first step here, we're going to say the Golgi packages the molecules into vesicles. So that's the first step of exocytosis. So these molecules that you see here in pink, those are the molecules packaged into our vesicle here. So we know our Golgi is responsible for sorting packaging and shipping our molecules. In step 2, that's right here, so this image, again, imagine this a few minutes later as the vesicle moves towards the plasma membrane and it's going to bump into it. So step 2, vesicle begins to fuse with plasma membrane and membrane proteins reorganize lipids for fusion. So this is where the energy comes in, the need for ATP energy. In order to reorganize here, you need to input a little bit of energy to these proteins that help reorganize and allow the vesicle to fuse with the plasma membrane. Also, on the movement towards the plasma membrane, ATP energy is used to move vesicles along the cytoskeleton as well. All right, finally, step 3. Molecules are released. Oops. Let's try that again. Released to destination. So here we finally get the molecules where we want. They have now exited the cell. Now, a few examples of this. So let's go ahead and do some examples down here. So our first example, number 1, of exocytosis, how it's used in your body. A huge one will be the movement of hormones out of cells. So we're going to say for our first example, transmission of hormones into the bloodstream from endocrine glands. Okay. So transmission of hormones into the bloodstream from endocrine glands. Endocrine glands, you're going to find several glands throughout your whole body. Each one is going to produce or store different types of hormones depending on what the body needs them for. So you have several in your head. You have the pituitary, hypothalamus, pineal gland. You have some in the neck region, the thyroid, parathyroid, in your chest and your thoracic area, which include like the pancreas, adrenal glands. And then of course you have even the endocrine glands that are part of sex, which would be the testes and the ovaries. So in order to release these hormones, cells will actually get signals that tell them to release these packaged hormones that are in these vesicles out into the bloodstream, as you would see here. So if this was an endocrine cell from an endocrine gland, it would release the vesicle to the plasma membrane, and then the hormones, so if you can imagine these would be hormones in this case, would be released into the bloodstream, and then they can flow out and do their job. All right, second example. I'm running out of room here. Let's go ahead and do it right here. So example 2, and this is a big one. This is often used when we're learning about exocytosis, and that's the involvement of exocytosis in transmitting signals throughout your whole body. Now, in order for your neurons, the cells of your nervous system, to send information through your body, it uses an electric signal, and it also uses a chemical called neurotransmitters. Electric signals flow through the neuron. So I'm going to draw a not so great picture of a neuron here, but you probably remember seeing this. So you have a neuron that has an interesting shape that looks like this. So this is one single neuron, one single nerve cell. And in order to send information, it sends an electric signal through the neuron. However, when the signal gets to the end, there's actually a space between one neuron and the next. So this next neuron here doesn't actually touch this neuron. So there's a little space, if you remember, it's called a synapse. When the electricity reaches the end of the axon here, we call them the axon terminals, the electricity can't just jump the gap. Electricity doesn't do very good jumping gaps. It won't be very focused or directed. So here, once the electric signal reaches the axon terminals, it changes to a chemical signal called neurotransmitters. Well, in order for the neurotransmitters to be released and float across the synapse to hit the next neuron here, that's where exocytosis is going to come in. So as soon as the electric signal reaches the axon terminals, the signal causes the axon terminals to release the neurotransmitters in vesicles towards the plasma membrane and then get released to the gap, the synapse in between. They'll float across, hit the dendrites is what we call them, of the next neuron, and then start the electric signal all over again. So our example 2 is going to be neurotransmitters And these are chemicals that axon terminals release to send a signal across the synapse to the next neuron. I had to squeeze that in there. All right, so that is our example number 2. Okay. All right, so make sure you have all of your notes complete here and ready. And if you have any questions, make sure you have those questions ready for next class.